Click. Here we go. All right. Well, welcome everyone to our February installment of the Data Portal Lunch Break series with the Consortium of California Herbaria. Today, we're going to be talking about some relatively new developments with the Linked Resources tab, which you may or may not have dabbled in so far. So today, we're going to just kind of take a step back first and talk about kind of the origin of the Linked Resources tab and why it's important. And one of those main things is this concept called the extended specimen concept. It's been a relatively new concept that's come up in the last few years that's been really pertinent to how the portal has evolved. And then we're going to get into the nitty-gritty about what the Linked Resources tab is and how you can use it, because it has changed relatively recently. In the Linked Resources tab, you can put associated occurrences, which is like other specimens or observations or taxa that are associated with whatever occurrence you're working on. You can also associate checklists. You can associate duplicate specimens, which are just kind of a different version of an associated occurrence. And then you can associate genetic resources, so links to hopefully GenBank or other places bold where you may have put DNA sequences. And then, of course, we'll open it up for any questions you might have. Okay, so the extended specimen concept was apparently coined in, I think, about 2017 with this paper that was about, I think, ornithological specimens. It might have actually been a book. And the idea is that a single specimen extends beyond the singular object to potentially limitless additional physical preparations and digital resources of a specimen. So the idea is that you have your physical specimen. So in our case, we have all these plants on sheets, but there's a lot of other data associated with that plant specimen that might end up not being on the physical specimen. So in our case, we have phenological data that we're scoring in association with these herbarium specimens, and those data aren't really on the sheet, they're extracted from the sheet. And so that's kind of an extension of the data that is physically in our collections. Same thing with tissue samples and preparations and genetic data. Those are all considered extensions of our data. So you might have seen this, this figure coming about. This is from a report by Tiers et al., who are forming the Extended Specimen Network, which is kind of like a, from my understanding, a network and think tank of people who are thinking about how we can make the best infrastructures and the best digitization practices to support the extended specimen concept. So the ability to link specimen data to all the other types of data that might exist out there. So beyond our physical specimens, we have the digital record, the media, so like the image of the specimen, but then there are so many different extensions, including field notes, morphometric data, descriptions, distributions, traits, things that are associated with the taxon, things that are associated with that specific specimen. So lots of ways that we can think about extending the data that we have in our collections. So I'm talking about all of this because it relates to the Linked Resources tab, because the things that are in here, you could consider extensions. So we have things that are gene sequences. We might have other field images that are stored as different occurrences. Stuff like that can be in the same, or can be considered extensions of our specimen. Okay, so without further ado, let's just hop into what this looks like. So I'm just going to go into my collection. So I've logged in. I go to my profile, occurrence management, and then I'm going to go to the collection that I'll be working in today, which will be the Cal Poly Herbarium. And so to view the linked resources, you can just look at any specimen. So let's just look at this random yarrow right here. And it's just one of these tabs in the top of the occurrence editor. So we have linked resources right here. So in linked resources, you can do several different things. And one of the ones that is perhaps the most confusing, but also one of the strongest, is this associated occurrences box. So right now you can't see anything there, but if I were to click this green plus sign, I could create a new association. And that association can be one of 3 different types of things. And you can kind of see the organization by how many boxes there are. So here's the create a new association. And there are 3 types. There's an occurrence within system. And the system we're talking about here is CCH2. You can do a occurrence to an external system. So that would be a different Symbiota portal or a completely different like Forest Service database or some other database. Or you can make a reference to another scientific name. So this is similar to if you entered a taxon in the associated taxa field. You can also do that in the linked resources as a observational reference. So if I wanted to link this to a specific occurrence within my system, so let's say that this specimen here, which is OBI 141400, is closely related to, let's say it's the same population. Actually, that would just be a duplicate. Let's say that this is a branch of another tree. So, I mean, I know it's not because it's a yarrow, but bear with me here. Say that it's part of this other specimen. So this one is 165628. Then in my original record, I could go into linked resources, add occurrence, and then I can search for the occurrence that I want to link this to by putting that in the identifier. And then I'm going to say, okay, that's the catalog number. So I'm going to look for anything with that as the catalog number. You can also select which collection you think it's going to be in. You just do a little search right here and it'll say, hey, do you mean this record right here? And let's say, yeah, that's the one I mean, because it's the same, it's the one that I expect it to be. So now that we've decided whether it's an occurrence within system or an external occurrence or an observational occurrence or reference, I've selected this one. Now we have to, or we don't have to, but we can define what the relationship of this specimen with the other specimen is. So we can say, oh, the relationship of my specimen with this occurrence is that my specimen is part of this other specimen. You can even have subtypes and you can say, I observed or I have in my living collection this relationship. So I would probably say like human observation, like I viewed that this specimen was part of this other one. It was a branch of this. If it was a like a lichen or something, and you're saying, I found this lichen on this tree, and you took a collection of both the lichen and of the tree, then you could also designate a location on the host, which is pretty cool. So this is especially useful for when we're talking about associated occurrences that have to do with ecological interactions, because you can say host of or has host or sibling of, things like that. And so you could say, oh, it had the host of this specific specimen, so that we know that those two were not only like in the same place at the same time, but one was hosting the other. So then I could click create association, and that will show right here. So my specimen is part of this other specimen here, which is pretty neat. So also in here, you can, so if I wanted to just do like one taxon, you can put a verbatim scientific name. You can say, oh, at the same time, I also found this scientific name here. And then you could click create association. Oops, I need to set a relationship. Ecologically occurs with. Boom. So you can do this. However, we're normally used to doing it just here in the associated taxa, and I'm not telling you to change your ways and put them in there because that would take way longer. But there may come a day where this linked resources is automatically populated from the associated taxa field. That would be cool. I'm not making any promises, but that's kind of the way that data integration and mobilization is going, because that's going to enable people to better understand the relationships between taxa in the future. So could be a development of the future. So other things that you can do in this tab is you can add your taxon or your specimen to a checklist. So if you're managing a checklist, let's just open up a checklist here. Let's just say here. Usually you will add taxa to checklists and add vouchers to checklists through the tools that are here. So like managing linked vouchers, and you can add vouchers through here. But if you know specifically which specimens you want to put into your checklist, you can just do that right here. So if you click on this, it will give you a list of any of the checklists that you have editing permissions for. And so you could say, I want to put this in this checklist, and then it will show up in the future that that's part of that checklist. This was useful for us because we have a Plants of San Luis Obispo County synoptic collection that we keep in some like other cabinets. And so we ended up creating a checklist for that whole synoptic collection, and then just linked all of those specimens to the checklist there. So that way we know that they're all kind of linked together in one project. Another thing you can do in the linked resources tab is look for duplicates. So this is something that you can do in batch. If you go to back to your data editor control panel, and if you click duplicate clustering, and if you click batch linked specimen duplicates, it will do this on a broader scale across your whole collection. It will look for all specimens that have the same collector name, number, and date, and then it will batch auto link them. But if you didn't want to run that whole thing, you just wanted to look for duplicates for this one specimen, you could also do that here. It'll just search based off of last name, number, and date. And if it doesn't find any, obviously it's not going to give you any, but that's how you could link duplicates from there. I will get to your questions in just a moment. I just want to finish up this genetic resources tab. So in the genetic resources, this is where you might want to put information, again, like from GenBank or Barcode of Life or wherever you're depositing your specimens. You would want to put the name that you've given to that sequence. You want to put the locus and then the URL, hopefully a stable URL where you can access that genetic sequence. So if you have deposited them on NCBI, for example, that's what you want to put there is the NCBI URL. Okay, Dawn asks, will links resources show in the public display of the specimen? I believe so. So if I go up to public display, yes, you'll see there's a linked resources tab, and then you will be able to see checklist relationships and data linkages. But I don't actually think that you can see all occurrence relationships. That will probably change in the future because this is very useful data that ecologists and such will want to be using. But I think right now it's not necessarily displayed. Okay, so those are the basics of the linked resources tab. They're relatively straightforward, except for this kind of mess that we've talked through. And so if you have any questions about this tool, I would love to hear them. And one important thing to note is that because this is so new, there are likely many other ways that it could be used. So, batch additions of linkages and linking to new resources or different resources. And those will probably be developed in the future, perhaps with future grants, perhaps with the sustaining grant that IDIG-Bio has, but this is just kind of how you can use this right now and what the idea is behind that.
I have a quick question. Maybe I missed it, but I was wondering if, like, for example, for the genetic stuff, basically, because this is what I do, is there like any place to link maybe a scientific article where those genetic resources were used?
You might put that in the notes, but no, there isn't a real robust place to put publications. That would be a great addition for the linked resources tab, though, because then you can actually see what papers have been using.
That's an interesting question, though, because like on the determinations tab, there's a reference section, so you can add a reference for a determination. And then sometimes annotations are just that specimen was used for genetic sampling for this study. And so maybe the most appropriate place for that is in the determinations table. But it's an interesting, like, those two things are kind of like crossing over now, the determination and the occurrence.
Yeah, I just think that we are using more and more like these specimens for research. And maybe it will be cool, like for any kind of research, phenological research, genetic research, to kind of like just have that, what has been done with this. And I don't know, it also increases the visibility of it. Like, hey, we use it.
For sure, it would be so cool. And eventually, I think people are looking to do this, that when you cite like a data set, then you'll be able to push back the citation to the original data. I mean, that's many years in the future, I think, but that's the dream.
I will say that notes section on the genetic resources has a character limitation, so you can't really put like a billion words in your semicolon title if you're doing like a molecular paper linkage. So like the way that I've been handling it is doing checklists for papers that are coming in for kind of draft forms, but then I get like little mini projects. And so it would be nice to be able to add like the actual publication. Yeah, that's one thing.
We could probably expand that character limit. It's probably it's like 200 right now or something. Or is it even less? You're muted if you're talking.
Oh, I don't actually know. I just know that I tried to put in something that was longer than it allowed, and it just stopped my typing.
Yeah, I think we could fairly easily change that in the database. Any other questions or comments? All right, relatively short session today. I'm going to stop. Oh, Dawn has a question.
Yeah, sorry, I needed to unmute myself. Probably just a question on usage, because in the original, when you talked about it, you were talked about also linking field images, but some of our botanists are not only linking images of the actual vouchers, but they're actually in the images section adding the actual image from, out in the when they collected it, picture, So any guidance one way or the other in terms of usage?
I think that still belongs in the images tab because that is an image of the specimen in a different capacity. There are ways that you can tag images, and you've probably seen this. So if you go into the images tab for a specimen, and if you upload an image, there are all these tags that you can use, and one of them can be field image of habitat. So that could be something that you could use to kind of indicate that it's a field image.
Okay. Yeah, I'd have to go back and check exactly what a couple of them, I don't know what they've selected, but okay.
And let me know if you need anything batch selected, like if you're like, oh my gosh, we forgot to do it for 150 of these, and now I'm gonna have to go and individually do it. I can do that on the back end, so let me know.
Sadly, we're still working with just imaging the specimens. We'd like to get all of our vouchers imaged and uploaded. That would be the first step.
Is there a way
Pandemic
to
sort of brought that to a halt.
Is there a way to batch upload the skeletal records for images? So to create the file, like you still have to link the actual uploaded images for something like an observational like group, the way that we upload records for like physical specimens, or is it the same thing?
So if you have images of like the habitat and you want to link them to the images of the specimen, is that what you're saying? Like how can you batch do that?
I mean, if it's just observational photos that we got like a giant thing from an entomologist, and he hates bugs now, and so now he's taking pictures of flowers, which is great, but he wants those uploaded in a way that's usable for other people. So I can do that individually with Cal Photos one at a time, but if there's a way to batch upload it with the Excel spreadsheet or the CSV file into CCH2, it would make my life a billion times easier.
You know, maybe this is an unpopular opinion, but I would just put them in iNaturalist because they are observations. We have the capacity to batch upload and create observational collections in CCH2, but the bread and butter of CCH2 really is specimens. And so putting those into iNaturalist, you're going to be able to reach the stakeholders who are interested in observational collections, not just specimens. And then those iNaturalist observations, once they are verified and identified, will go into CalFlora automatically.
Okay, thanks.
All right, I'm going to stop